Hi biologists, let's start by looking at the learning objectives for this lesson. At the end of this section and following the biology syllabus, you should be able to identify and know the function of the mitochondrion, the chloroplast, the nucleus, nuclear pores, the ribosome and DNA. What does this actually mean? What are we trying to understand? Well, it's straightforward enough. You have to be able to identify and know the function of the following cell parts. You have to be able to identify and know the function of mitochondria, chloroplasts, the nucleus, nuclear pores, ribosomes, and finally DNA. Before we can identify and know the functions of various cell parts, which is the objective of this lesson, we need to revise some ideas we met before. Remember, an organelle is a structure in a cell. They are usually colourless and have to be stained in order to be seen under the microscope. We also have to understand that ultrastructure is the detail of a structure as can be seen using the electron microscope, which is very powerful. Now, let's have a look at a mitochondrion. The structure of a mitochondrion reminds me of a piece of corrugated cardboard with its smooth outer surface and its folded inner surface. The mitochondrion is quite similar, to my mind, because it has a smooth outer membrane and a folded inner membrane. The individual little folds are called cristae. The centre of the mitochondrion is called a lumen, and the mitochondrion has a loop of DNA. This is a vital point because a mitochondrion, along with a chloroplast, are the only two organelles that have their own DNA outside of the nucleus of a cell. So in a nutshell, how would we identify a mitochondrion by its structure? Well, it's surrounded by a double membrane, a smooth outer membrane, a folded inner membrane. The infoldings are called cristae. It has a loop of DNA. The function of the mitochondrion is to supply energy to the cell. In other words, they carry out respiration, getting energy from food. Active cells have a lot of mitochondria. This makes sense because active cells carry out a lot of chemical reactions. Muscle, brain, and liver cells in humans have a lot of mitochondria, plural. Meristematic cells in plants actively carrying out mitosis or actively carrying out cell division also have a lot of mitochondria. Inactive cells that don't do many chemical reactions have fewer mitochondria. Fat storage cells in humans have few mitochondria. In other words, the cells that make up adipose tissue. The ground tissue in plants, whose function is to store food in the form of starch, they also have few mitochondria. Be able to recognize the electron micrograph, a photograph of a mitochondrion under the electron microscope. We recognize it because of its oval, tic-tac shape and the infoldings or the cristae. What about identifying a chloroplast, which is another objective of this lesson? Now this time, chloroplasts are the structure, S for structure, S in chloroplast. They are the structure that contains the green colour called chlorophyll. Now this time, chloroplasts remind me of creme eggs. Basically, you have the outer layer of tin foil, then the layer of chocolate. So we have a smooth outer membrane and we have a smooth inner membrane. Just like the egg is filled with goo, the chloroplast is filled with a watery substance called stroma. Running across the stroma are a series of membranes which can be folded over on top of one another. These folds are called granums. Strictly speaking, grana for plural. And it is in the granum that you find the chlorophyll, the green colour or the green pigment. Chloroplasts also have starch grains. This is logical enough when you think about it because their function is to make food for the plant, so the food has to be stored in the form of starch. As we've said already, chloroplasts have a loop of DNA. Again, we need to highlight the fact that the chloroplast and the mitochondrion are the only two organelles which have their own DNA. What is the structure of a chloroplast? Well, it's surrounded by a double membrane, a smooth outer membrane and a smooth inner membrane. There are stacks of membranes within its structure called grana, plural, and these contain chlorophyll. The chloroplast is filled with a dense fluid called stroma. There is a loop of DNA and starch grains in the stroma. The function of the chloroplast is to carry out photosynthesis. Remember, photosynthesis is the way plants use light to make food. Chloroplasts are only present in photosynthetic green cells, like most normal plant cells or algae, which would be seaweed or pond scum. Recall that chloroplasts are not actually found in onion cells, otherwise onions would be green. Chloroplasts are not present in animal cells or bacterial cells. Again, be able to recognize, identify the chloroplast in an electron micrograph. We would recognize the chloroplast because of its characteristic oval shape, the presence of the starch grains, the membranes running across the stroma, and the folds of membranes called the grana. How would we identify a nucleus? I find it useful to imagine a ping pong ball punched with holes containing a jelly and a couple of marbles inside. The ping pong ball, the white plastic, is representing the double nuclear membrane. The holes, obviously, are representing the nuclear pores. The nuclear pores allow chemicals in and out of the nucleus. The jelly is called nucleoplasm. The word plasm should be ringing a bell. It is a jelly within a nucleus, in the same way that cytoplasm is the jelly within a cell. In the nucleus, you often find nucleoli, plural, or a nucleolus. The nucleolus makes the ribosomes for the cell. In the nucleoplasm, you also find chromatin. Chromatin is an invisible material. IN in chromatin, IN in invisible. And the chromatin is made of a chemical called DNA, and when you can't see it, it is referred to as chromatin, and when you can see the DNA, it is referred to as chromosomes. So in a nutshell, what is the structure of the nucleus? Well, it's surrounded by a double membrane. The membrane contains many nuclear pores. There is hereditary material within the nucleus called chromatin, which is invisible. It contains a nucleolus or more. The nucleolus makes ribosomes. The function of the nucleus is to control and regulate the cell's activities, for example, growth and metabolism. It controls the chemical reactions in the cell. You could say it is the control center of the cell. Be able to recognize an electron micrograph of the nucleus. We would recognize the nucleus because of its round shape and the darker round staining in the center, representing the nucleolus. How would we identify the nuclear pores? Well, the nuclear pores are small holes in the nuclear envelope or double membrane. And the function of the nuclear pores is to allow chemicals to pass in and out of the nucleus. Basically, they allow communication between the nucleus and the cytoplasm. For those of you able for a bit more, it might be no harm to take on the names of the chemicals that pass in and out of the nucleus. One chemical is messenger RNA. This passes out of the nucleus during protein synthesis. Another chemical is ATP and nucleotides. These chemicals pass in and out from the cytoplasm. How would we identify DNA? Well, DNA is a complicated chemical. The bad news is we have to know what the letters stand for. They stand for deoxyribo, nucleic, because it is in the nucleus, acid. DNA forms thread-like structures called chromosomes. Genes are sections of DNA found on the chromosome. When the DNA is invisible, it forms thin, elongated strands called chromatin, which is invisible. So in a nutshell, the structure of DNA is that DNA and protein forms strands or structures called chromosomes. These are visible, coloured bodies. Chromo means coloured, some means body, like somebody. Get it? Genes are found on chromosomes. Genes are actually sections of DNA, and chromosomes are usually in the form of fine threads called chromatin, which are invisible. The function of DNA is to regulate the making of protein in the cell. As a matter of interest, genes are actually responsible for regulating protein synthesis. Genes, don't forget, are sections of DNA, and they actually contain the code for making protein. Genes are the units of inheritance because we inherit them from our parents. How would we identify the ribosomes in a cell? Well, ribosomes are tiny organelles which are free in the cytoplasm or can be attached to the endoplasmic reticulum. They are made of RNA. Again, the bad news is we need to know what the letters RNA stand for. They stand for ribo, nucleic, because it's in the nucleus, acid, A for acid, and protein. This RNA can sometimes be called ribosomal RNA. Ribosomes consist of two different sized subunits, rather like burger buns. What is the function of the ribosomes? Well, the ribosomes make protein. They carry out protein synthesis. They actually link amino acids together. This process is called translation. Finally, to summarize, what are the functions of the cell components? As a little heads up here, we are dealing with functions. Don't forget, we must use verbs. We must use action words. If we look at all the functions in this table, we can see that they're all action words. carry, controls, allows, regulates, makes. So the function of the mitochondrion is to carry out respiration, supply energy to the cell. The function of the chloroplast is to carry out photosynthesis. In other words, use light to make food for the plant. The nucleus controls and regulates the cell's activities, whereas the nuclear pores allow communication between the nucleus and the cytoplasm. They actually allow messenger RNA to leave the nucleus. The DNA regulates the making of protein in the cell, and the ribosomes make the protein or link the amino acids together. And there you have it. Now that we have reached the end of our lesson, have we achieved our objective? Can you identify and do you know the function of the mitochondrion, chloroplast, nucleus, nuclear pores, ribosome and DNA?